Now that you've got the basics of how protein synthesis works, you have your transcription from DNA into mRNA, and then the mRNA is translocated, so it leaves the nucleus and moves out to the cytoplasm, and then it's translated by the ribosomes into amino acids, and then that gets folded and whatnot, and you end up with a final protein. That's your tertiary or quaternary structure. So hopefully you've got that basic idea down with the terms transcription and translation. But now I need to get into the details of focus on the steps that actually happen here in transcription, and then in the next part of the lesson, the actual details for translation as well. So let's dive into it. Now, of course, the goal of transcription is that the DNA template strand, not the coding strand, but the non-coding strand, gets copied into messenger RNA or mRNA. It's important to note some differences between RNA and DNA. Obviously, they both carry genetic information, but RNA has a different structure, and this actually ends up being a really good test question. We focus on the differences in structures and similarities, actually. That's also part of a good test question. Basically, you should already know this, but it contains a ribose sugar instead of deoxyribose. In other words, at the 2' carbon, it has the full OH group, whereas the deoxyribose only has the H, so deoxy is missing an oxygen. It contains uracil instead of thymine. That should also be not news to you. It still pairs with adenine, though. It's a short, single-stranded strand instead of being double-stranded. That saves a lot of energy when we're copying. And the DNA can move between the nucleus and the cytoplasm, but the DNA cannot. Now, this last one is a location difference. It is not a structural difference. So if you're giving me similarities and differences in structure, it can be talking about these three first things, but not this last one. That's a location difference. Okay, so be careful when you're answering that on your test question. The mRNA is going to be complementary to that original DNA template strand. So a reminder that the DNA template will run 3' to 5' because it's in a nonsense direction, so that the actual mRNA, which does make sense, can be in the 5' to 3' direction. Okay, we call that a primary mRNA transcript because it has not been modified yet, and I'll show you what we mean when we talk about modification. But before we get there, just a reminder that when you're doing your start and your stop codons, you always want to read it in the mRNA state when you're jumping into the amino acid chain. You don't need to care about the tRNA anticodon. So that codon chart that I showed you last day is a direct link from mRNA straight to the amino acid. So I can take a look at this mRNA right here and know already that, hey, AUG, that's methionine. I don't need to flip it to the anticodon of the tRNA and then to the thing. I can just jump straight from mRNA. Okay, said that enough times. There are 3 different types of RNA molecules. You have your messenger mRNA, which is a longer strand. Of course, the length is going to depend on the size of the gene that you're copying. And this is what gets translated into protein by the ribosomes. Then you have the tRNA, or transfer RNA. That's these little units here. And you can see better than the last day's example that this is a long strand of RNA that's just been folded in on itself, and it has complementary pairs opposite one another so that they can hydrogen bond. Same on this section, same on this section, same on this section. At the 5' end, it has a phosphate group, just like every other DNA or RNA molecule. And at the 3' end, it has an open hydroxyl group. This hydroxyl group is where we're going to end up adding in the amino acid. Okay? And then, of course, at the other far end, you've got the anticodon, which would be opposite to whatever's going on with the RNA codon, the mRNA codon. So anticodon just meaning opposite. Okay, it's going to match the mRNA codon. UAU would be the opposite of AUA, and UAU is the code for tyrosine. It's one of the 20 amino acids. Okay, the final type is ribosomal RNA or rRNA. We don't do much beyond just mentioning that it exists because that's beyond the scope of our study in grade 12. But the basic idea is that part of the ribosome is made up of ribosomal RNA. It's protein stuck with that. And that helps us to actually read the mRNA. And it also allows us to actually bring these tRNAs in. Now, that can be all different lengths. But again, we're not going to focus on that. Just know that it exists. So you have mRNA and tRNA are the RNAs that we really care about. rRNA, you just have to know it exists. Let's move on. Let's get into the details for transcription. We're going to have initiation, which just means starting. Then we're going to have elongation, which just means to make the longer chain. And then we're going to terminate that chain, and we'll talk about that as we get there. Now, the RNA polymerase is going to do the job of opening the double helix. I don't need the helicase in this case necessarily. The RNA polymerase can do that on its own. And it's going to bind to the DNA at something called the promoter region. We also call this the TATA box because this promoter region has tons of A's and T's. So TATA is just a cute little way of saying lots of A's and T's. Now, why would we care that the RNA polymerase likes to bind to this region? Why A's and T's? Why not C's and G's? Well, remember that there's only 2 hydrogen bonds between A's and T's, and it takes way less energy to break 2 of these bonds than it would be if the place was rich in G and C because there's 3 hydrogen bonds. Please know this as an answer for a test question. The promoter region is usually rich in A's and T's, takes much less energy to break the 2 H bonds between the A and the T than the 3 H between, or the hydrogen bonds between the G and the C. Notice that the promoter region does not get transcribed. Okay, so here would be an example of, let's say this is our promoter region here. RNA polymerase attaches and it reads along until it sees this area. It pops this open, but it doesn't start to add in any RNA nucleotides until it reads the start codon. Then it can begin to add things in. So you're still starting at the start with the mRNA. But the TATA box is in a location that we call upstream. See, this is the 5' end and this is the 3' end of the coding strand, not the anticoding, but the coding strand. Upstream means to the left or to the 5' end. So you always have the promoter here because the RNA polymerase always starts on the left and then moves towards the right as it's going. Okay, so RNA polymerase again is the enzyme, the ASE enzyme that makes the polymer, which is lots of nucleotides joined together, and the polymer is made out of RNA. Okay, so TATA box, memorize that. Promoter region, TATA box. You need to be able to interchangeably use those terms. Okay, so again, you're going to find the start codon, and then you're going to begin to use the template strand to go ahead and copy that little section of DNA that you want to make into mRNA. Okay? Again, notice that you don't use the sense or coding strand. That's non-template. You're only using the nonsense or the antisense non-coding strand as your template. Another key thing, as we elongate, keep in mind that the RNA polymerase is going to use the DNA template strand, which would be this strand right here. It's going to use that strand to add the complementary RNA nucleotides in, but I don't need an RNA primer the way I did with DNA replication. That's because it's all going to be RNA. So I can just start right away and I can just run. It's a much faster process. I don't have to worry about little Okazaki fragments or anything like that because I'm only using the one strand anyway, and it's going to run in the 5' to 3' direction continuously, so it's going to be fine. All right, so no RNA primer required here. I don't need any of that extra junk. So I don't need any primase or anything like that. RNA polymerase can do its job all on its own. And again, we're going to build it 5' to 3'. Okay? So elongation just means you're going to continue to open your transcription bubble. So as the RNA polymerase moves along, it's popping hydrogen bonds and they're sealing up behind it. I don't need to use single-stranded binding proteins because I've only got a very short section open, and the RNA polymerase is able to keep it open just long enough for what it needs to do to copy, and it seals up as the RNA polymerase moves along. The DNA gets rewound, it gets put back together, and the mRNA starts growing out as a chain along there. Okay? Eventually, your RNA polymerase is going to reach a codon that doesn't really code for anything. And when it reaches a termination sequence there, it knows to fall off. It knows that it's the end of the gene. And then the RNA polymerase will fall away, the DNA will be rewound, and this mRNA will now be free to translocate out of the nucleus into the cytoplasm where the ribosomes are. Okay? And then finally, you have termination. That just means to end. RNA polymerase passes the end stop signal. You get the terminator sequence. Like I just said, mRNA gets modified, though. This is the key thing that's different. mRNA is going to get modified before it leaves the nucleus. Here's how it gets modified. Okay? There are 3 things that are going to happen in eukaryotes. This is different if you're talking about bacteria, but in eukaryotes, there are 3 modifications. We call them post-transcriptional because they're after you've transcribed the mRNA, and then you modify it. Okay? Post-transcriptional. This is before it leaves the nucleus. First thing you're going to do is to protect the 5' end. Remember that there's a phosphate at the 5'. So you add a little cap. It's called a 7-methylguanosine. You can see a picture of it if you look into your textbook. I'm not too worried about you seeing that at this moment. And the reason that we want to add that cap is that it's going to protect that mRNA strand from being eaten from this side. When it leaves the nucleus and it goes out into the harsh world of the cytoplasm, there are enzymes out there that are designed to destroy and recycle nucleic acids. So you really don't want this guy just free floating. So you have that little 5' 7-methylguanosine attached, and so that you're going to protect the mRNA for a little bit longer from being digested by a nuclease, which would digest nucleotides, or a phosphatase, which would start digesting from the 5' end. Okay, so again, just cap on the end there to protect it. On the other end, we'll have a poly-A tail, it's called. Poly-A tail because it's literally just a whack of adenine nucleotides that get added. Sometimes they can be up to 200 adenines just attached there. Now keep in mind, these are ribonucleotides, so they've got the ribose sugar, not deoxyribose. But they're added as RNAs, nucleotides all the way along. And that again is to protect it from digestion, because you're not going to necessarily go and bind right in the middle and chop something, although there are enzymes that can do that. But this will slow down the digestion so that I can use this mRNA strand for much longer. Okay, so I'm going to first cap it with a 5' cap, then add the protective 3' end poly-A tail. There's another thing that happens as well. Now we mentioned that even along a gene, there are sections that do not actually code for anything. Those sections are called introns. Okay, introns are useless for actually making a protein. It's only the exon sections that are actually going to code. So what we need to do before we leave the nucleus is to chop out the introns and put the exons side by side. You see that in this picture here. So you have the 5' cap, you have your poly-A tail, and you can just cut out these sections. This is cut out by something called a spliceosome. It's an enzyme that recognizes the end of exon 2 and the beginning of exon 3, and it's able to chop that out. Let me switch back to the animation we saw yesterday so you can see it in action. So here we see again the transcription of the mRNA. We've got the free-floating RNA nucleotides being grabbed by this RNA polymerase that's in behind here. It's matching it up to the template strand, and you can see that the mRNA strand is complementary to the template strand that is poking up there. Now here we've got our mRNA transcript, but we haven't finished editing it. We're going to have to add on that 5' cap, you see that up here on the left there, and then the poly-A tail on the 3' end. And then we've got these introns that are going to need to get cut out. This little guy here is called a spliceosome. Splicing means to cut. You will see that the exons get folded in towards each other, and then the spliceosome will chop out that intron right at that center there, and the intron will remain inside the nucleus. So when you're thinking intron, think inside nucleus. When you're thinking exon, think exits the nucleus. So they get spliced together there, and now I've got myself something called the mRNA transcript. So primary transcript was before I modified it. mRNA transcript is what it's called now. Now keep in mind, there's no quality control here. You don't have any extra checks or balances to make sure that the script is correct, just like there would be in DNA replication, and that could technically lead to incorrect proteins. But the RNA is a temporary molecule. It's only going to be used so many times, and then it's going to be degraded. So it's not a permanent mistake in proteins. Plus, if you remember what I showed you yesterday, when you're looking at the codon chart, the third base in the codon triplet is way less important than the first or second. So if there's a mistake in the third, you may not even change the amino acid. In most cases, the amino acid stays exactly the same. So there might not even be a problem. So no quality control the way that we have it in DNA, but it doesn't lead to the same issues. And those proteins might end up being temporary. It's not a permanent mutation. Now, there is this idea of alternative splicing I want to mention quickly. There are approximately 100,000 proteins in the human body, but only 25,000 genes-ish that we found. The cells then must have the ability to choose which exons can be included in the mRNA transcript to allow that single gene to actually code for multiple proteins. So in other words, let's say for one particular protein product, I need exon 1, 2, and 3 to make my product, because this has the code that has all the amino acids that I need in order. But for another protein, maybe I don't need this exon in the middle at all. Maybe I just need 1 and 3. So when I chop out these 2 introns, I can take that exon with me and just have 1 and 3 as being the final product, or 1 and 2, or 2 and 3, or whatever the combination might be. In that way, we can have one gene encoding for multiple different versions of proteins. So similar sections within the proteins, but an overall very different final result, which means it could have a completely different function. So that is actually another amazing level of information stored within the DNA that the actual cell would know when to cut out just an intron and when to cut out another exon as well. So again, that's called alternative splicing, and it's actually a pretty exciting, newer concept than some of the other stuff that we know. Let's flip over now to talk about the translation detail. So just a reminder that translation means that you're taking that mRNA transcript that you've already created. It's already been brought and translocated out to the cytoplasm, and it's going to be read by these ribosomes to make ourselves a nice polypeptide chain. Okay? We have the same idea where we initiate, we start, then we elongate, and then we terminate. We have those fancy little words. So start, middle, end is really what it means. Now the ribosome is going to recognize the specific start codon. It's looking for that AUG that's written there, and it's always going to put methionine in that spot. AUG is the code for methionine. It's going to bind to that site, and then the ribosome is going to move over 3 nucleotides at a time. In other words, 1 codon at a time, and for every 3 nucleotides, it's going to read it and deliver the right complementary tRNA, which has the amino acid attached. Here's what ribosomes actually sort of look like. They've got this larger folded protein. It's called 60S versus 40S just because of their size. It refers to their density when they get centrifuged. So this one's just the bigger unit and the smaller unit. But in the bigger unit, there are 2 very important sites. We have the P site, P for peptide, and we have the A or acceptor site, which I'll talk about again in a moment. Now, it's very important that the ribosome actually attaches to the correct end so that it reads the mRNA in the right direction, and it recognizes that 5' cap that we put on there, the 7-methylguanosine cap. It recognizes that it's located at this end, and it knows to clamp on in this direction so that it reads the mRNA correctly instead of reading it backwards. It's very important. So we've also got the moving or the translocation of the ribosome moving along there. It also moves in the 5' to 3' direction. Okay? And a new amino acid gets added in every time it reads a codon. Every time it sees the 3 nucleotides, it's going to add in the correct amino acid. We've got something called a reading frame. This is really important, and it's going to come up again when we talk about mutations. You have to start at the right codon, or the whole protein could be affected. Okay, so here's our AUG. This is where we should start, and we would add in a methionine, then CCA codes for proline, GAU for asparagine, so on and so forth, alanine, isoleucine, doesn't really matter. Okay, so moving on, you can see that these ones down here, if you start at the start, you get the right proper amino acid chain. If, however, the reading frame was shifted, let's say that the ribosome saw UGC. Well, if it saw UGC instead of AUG, this is the code for cysteine. That's very different than methionine. Pro and glon, very different. Methionine and asparagine, different. Proline, alanine, different. It would make a very, very different set of amino acids. And just a reminder again, changing one amino acid in a protein could be devastating to its function. Not always, but many times it can actually change the entire effectiveness of that protein. So the reading frame has to be correct. You have to start at the start and then run through. So it's very important that the ribosome is able to read. Okay, it has to be a literate one. Okay. Then the transfer RNA, just a reminder, they're going to have their little anticodon that's going to match up with one of the codons here. It's got the 5' end where it's got the phosphate, and then the open 3' end with the OH. This is where you attach the amino acid. When the amino acid is attached, we call that charged. And when there's no amino acid because it's already been delivered, we call that uncharged. So right now you're looking at an uncharged tRNA because there's no amino acid attached. Quickly talk about the Wobble hypothesis. It's a lowercase w because it's not a person's name. It's just the idea of literally something floating back and forth, wobbling. Okay. Now, there's only 20 amino acids that we require in the human body. But if you look at how many possible codons there are, because there's 3 positions for the bases, you have 4 possible bases in the first position, AUCG. Second position could be AUC or G. third position could be A, U, C, or G. So when we multiply those possibilities together, there's actually 64 different codes that could be 3 letters long if you had 4 letters per spot. Okay, but we only have 20 amino acids available. That's why there's multiple codons possible per amino acid. And if we were to flip back to our chart here again, you'd see that you have multiple codes available for proline. Remember, you have 6 possible for leucine. There are only 20 amino acids that I have to worry about. In fact, some of them don't even code for anything. These 2 here and that one there, they don't code for any amino acid. That's why they become stop codons. Why is that so great? Well, it means that you've got flexibility for errors. Remember that we are not proofreading this when we transcribe very well. We do a little bit, but very, very little. RNA polymerase just goes for it and transcribes that DNA into the mRNA. So there's a lot of flexibility then for errors when you can mess up a code in 1 or 2 positions and still end up with the right amino acid. So that's great. But again, there's only 1 code for methionine and actually only 1 for tryptophan as well. And the stop codons do not actually code for any amino acid whatsoever. And those are those 3 codons there that we just saw on the other page. Okay, so we start at the start. The start codon makes sure that the reading frame is correct. You don't want to start at the wrong thing and then start adding in all the wrong amino acids. So this little AUG, as the ribosome is reading along from the 5' end to the 3' end, so it's going to move in this direction, it suddenly sees the AUG, and that's the code for methionine. Notice that the methionine is able to go into the P site, P for peptide. It's called that because that's where the peptide bonds are made as the next one comes in. So methionine is the right shape. It's actually the only one that is actually able to enter the P site. Then you'll have another tRNA bring the next, so you have these 3 little letter code here, this little codon here. You'll have the next one come in. These guys will have their amino acids attached at the top, and then the ribosome will move to the right. And when it does that, the methionine tRNA that was sitting in the P site suddenly finds itself in the E site. I can say E for exit if you like. And then it breaks off and leaves the amino acid behind. And the one that would have been in the A site moves to the P site. And then it's free now to accept. That's why it's called A for acceptor, to bring in a new one. There's a better diagram down below that I quickly want to show you. So here we have the methionine attached to the tRNA, and it's matched up to the start codon. We've got an empty A site. And then in the next picture, you can see here's alanine has been brought in. Now it's sitting in the A site. But then you're going to see that this whole ribosome is going to shift over. It's already shifted over between this picture and this picture. It shifts over to the right. That pops out this tRNA, the one that was sitting here in the P site, the one that had methionine attached. That tRNA gets popped out. This one here, the alanine, gets moved to the new P site. And then a new isoleucine is able to come into the A site here. And you just have it moving along, moving along, moving along. You can see the amino acid chain growing. So when something is in the P site, it gets attached to whatever is in the A site as far as the amino acids go. And then eventually you get to the point where you have in the mRNA, you've got the stop codon listed there. It doesn't code for anything. The ribosome cannot grab any tRNA because one doesn't exist. So then this guy is now free to pop out of the P site. There's nothing added to the A site. So then the ribosome can just fall apart because it's empty now. And it's going to go fall apart and be used somewhere else. And in the meantime, we've created our nice new polypeptide chain. Again, methionine goes straight to the P site. And then all the other amino acids can go to the A site and get joined to the methionine. So then let's quickly chat about termination. Now we're going to stop at the stop, like we said. The ribosome falls off and the polypeptide gets let go, and it's free to go fold now and be finalized by either the endoplasmic reticulum or the Golgi body. But the polypeptide chain itself is not a functioning protein until it gets folded. Okay. Sometimes you have to add a sugar or a phosphate. It's called glycosylation or phosphorylation, when glycosylation means just to add the sugar. You might also cut off part of the chain or add other modifications. You could add, perhaps there's a metal ion that needs to be added in there, like the iron in the hemoglobin and whatnot. And even sometimes that methionine, that beginning one, is cut off. So whereas every peptide chain starts with methionine, it has to, because methionine is the only one that can go into the P site directly to start the whole elongation of the chain. It doesn't always have to be included because sometimes you're going to modify that that polypeptide chain before it's the fully functioning protein. And that, my friends, is the end of this lesson.